Good morning to Professor Dr. Edmund Sim Wei Hang. Today, my group will be demonstrating a program in bioinformatics called Basic Local Alignment Search Tool or BLAST. This is the outline of today's presentation. I, Narisa, will start off the presentation. First, what is BLAST? BLAST is a program that helps its users search for regions of local similarities between sequences they have inputted using the Smith-Waterman algorithm. With a heuristic approach, BLAST does a comparison between protein or nucleotide sequences against the database's sequences, not by its entirety, but finding short matches between two sequences. Then a statistical significance of the matches is calculated. Bidirectional extension of words occur during alignment, such as seen in figure 1. BLAST can be used in many ways. Firstly, to deduce evolutionary and functional relationship between sequences. Secondly, to assist in finding members of gene families.
This is a tutorial on how to utilize the BLAST software to compare between the same gene in two different organisms by performing pairwise alignment. We will be comparing the BRCA1 gene in dog and also red kangaroo. First, we have to determine the ascension number and also the FASTA sequence of the gene. So you can first access the NCBI homepage using any browser such as Chrome. So on the top right, on the top of the homepage, you can change this to nucleotide, and you can key in the name of the gene and also the name of the organism that we are interested in. So since we are interested in BRCA1 gene in dogs, so we will key in this as such and click on the search button, and it will appear a result page like this. So once you get this page, you can scroll down and select the first option of the search result. So click on this link. It will lead you to this page where you can see the information of this gene and obtain the ascension number over here. As for the FASTA sequence, you can click on this link over here and it will direct you to a page like this where you can obtain the FASTA, the sequence, the GIS gene FASTA format. So do the same steps for red kangaroo and you will obtain a page like this, get the ascension number and also the FASTA sequence from this link over here. Now that we have found both the ascension number and also the FASTA sequence for both organisms, we will now move on to the comparison of the two genes using pairwise alignment in BLAST. So go back to the NCBI homepage and click on the BLAST link under popular resource. So in the BLAST page, search for global alignment link and select it. And in the global alignment page, you can enter the ascension number of each gene respectively. So this one would be the ascension number for dogs and this one would be for red kangaroo. So click on the BLAST button and wait for a few seconds for the result page to appear. After a few seconds, the result page is as followed, and under the description box, you can click on this link to view the detailed results of the alignment. So as you can see, there is the numbers of identity and gaps between the two alignment. So out of 699 alignment sectors, there are only 30% identity between the two sequence, indicating that the BRCA1 gene in dogs and also red kangaroo, there are only minor similarities. And there are 62% of gaps between the two alignments, and the remaining 8% would be the mismatch pair between the two alignments.
BLAST has many advantages and limitations compared to software FASTA. First, both of them are algorithms that are used to compare primary biological sequences like nucleotide or amino-acid sequences. In terms of speed, BLAST is faster compared to FASTA because BLAST is used to search for more similar nucleotide and protein in sequences, while FASTA is used to search for similarity in less similar sequences. However, the accuracy of FASTA and BLAST tend to be different for protein and nucleic acid. For protein alignment, the accuracy in BLAST is higher than FASTA, while for nucleic acid, FASTA is more accurate than BLAST. It is because of the word size. Increasing the word size will increase the speed, but it also will cause loss of sensitivity. But there is an exception. Although the word size of protein in BLAST is more than FASTA, the accuracy of BLAST is still higher than FASTA. Lastly, gap between query and target is not allowed in BLAST, while it's allowed in FASTA. When aligning sequences, introducing gap in the sequence can allow an alignment algorithm to match more terms than a gapless alignment can.
Using the same characteristics, BLAST is compared to another bioinformatics software called Molecular Evolutionary Genetics Analysis or MEGA. MEGA is commonly used in analyzing proteins and DNA sequence data obtained from species and population. As for the speed, both BLAST and MEGA has the same speed, where MEGA is fast in searching the nucleotides and proteins in producing scoring and evolutionary relationships. In terms of sensitivity, BLAST is using the word size for both proteins and nucleotides, but for MEGA, percentage format will be used. In MEGA, the similarity threshold for both protein and nucleotide is low and high respectively. Nucleotides have roughly about 25% of similarity, while the sequence for proteins is 5%. The last comparison between BLAST and MEGA is the gap size in query sequence. When BLAST does not allow any gap between the query and target sequence, MEGA can include the gap size. Most of the times the gaps are ignored, but if the gaps are small, it can be included but it will be treated as missing data. That's all from us, thank you.